Since 1977, the Saba Lions Club has been a pillar of service and community care on the island. What began with 20 committed members has grown into a legacy of giving back, supporting those in need, and advancing the global mission of Lions Club International. From cultural events to signature service projects like Meals on Wheels and the Food Bank, the Saba Lions Club supports the elderly, disabled, and struggling families with compassion and consistency. Their health programs, like annual eye care support and glucose fairs, continue to promote wellness across the island. The club is also a champion for the environment, proudly maintaining an adopted hiking trail and backing local cleanup efforts to protect Saba's natural beauty. Several of its members have been honored with the prestigious Melvin Jones Fellow Award for their outstanding dedication, proof that the spirit of service is alive and well on Saba. The Saba Lions Club, serving with heart for the good of all.
Hello. Good evening, everyone. Good evening. Hello. It is a beautiful night on Saba. And I first want to just say thank you, Busy Bee and Hideaway, not our official sponsor tonight, but thank you, Rachel and your team, for hosting us this evening. Here we are, 22 years later. Yeah. It's our 22nd annual event, with the exception of 2020. But before we get into everything we want to speak about tonight, we need to officially open our event. So I want to hand the microphone for the last time, because he's going to tell you how many times he's done this, to those of you who don't know him. This is our Governor, Jonathan Johnson.
Thank you, Lynn. So as Lynn said, this is the 22nd year, and out of the 22 years, I've been able to do this 17 times. So this is my 17th time of opening See and Learn, and it's been a privilege and a pleasure. And See and Learn, as we all know, it's one full of learning filled, but also fun-filled activities for young and senior, I won't say old, but and all ages in between. But it's something that has been a real impulse to our island during the month of October. And it's something that, no one ever expected to grow in this manner and means, but something we're very proud of. And we're also happy and thankful for Lynn and her team, because with Lynn, there's a number of persons that assist her in making this all possible. So I'd also like to thank you all for being here this evening for the opening night. And I wish you all a fun-filled and learning-filled month. And with that, See and Learn 2025 is officially open. Thank you.
Thank you. And thank you to the Lions Club, who is supporting our live streaming this year. And I always feel bad because people thank me, but it's not just me. There's a whole lot of people who make this possible. We'll get into sponsors and such, but right now I'd like to introduce our team. We have our newest, well, before I get to our newest, we want to thank Yet, whose last day was day before yesterday. Our first education specialist, who made a big difference with our team, and we had a great year. And she's now moving on to the Saba Conservation Foundation, but that's okay, because we're going to hope to keep working together. So if you can stand up and take a bow there yet. And with that, our newest team member, our very experienced also teacher, our new education specialist. We're doing it with two people. So we have Debbie Verdalt and her partner Terry, who can't be here tonight, but this is Debbie, who many of you know already. And a few months ago, we hired a someone to help us grow our capacity of our culture sector of our foundation. So specifically working on the Adopt a Box project and taking it to another level together with Adam and doing audio components is I have to look at my notes because we gave her this title. Like, wait a minute, what did we tell you we are? So our Heritage and Development Coordinator is Miss Andrea Ellinger. And anybody who's been here before, or if you just got here, you've had a lot of communications because this event is absolutely not possible without our operational director, Miss Emily Melsack. Okay, so everybody knows we're going to get started, and we got a great evening, and we got a whole full month ahead. So I'm going to hope to use this and advance our slides accordingly. So here we go. So this event is also not made possible without a whole lot of money and things that make a month-long event happen. We used to, for 20-some years, talk about Prince Bernhard Culture Funds. We now call it CCG, which is the Caribbean Culture Fund, which is amazing and really helps us also with our live streaming promotion, all kinds of things. Public entity SEBA, we have a few representatives here tonight that also really make not the event but our foundation possible. And, whoops, different clicker. And all these sponsors. When we started this program in 2003, we had 4 sponsors. Sorry. Special seating considerations. And as you can see, we got a whole lot of sponsors now, so we don't go through them one by one anymore, except when we have a real technical problem, then we do the improv. But there's many people and many sponsors here tonight. Raise your hands from Tim and Larry and Sherry and all kinds of people, so thank you very much. But tonight, our speaker, so every night we will make a special mention for the accommodations who are very valuable sponsors because they really help us support having the expert scientists here for a week or more sometimes. So Althea's Cottage, Jeff, can you hear us up there? Thank you very much. Beautiful property taking care of a number of our scientists. Raffle tickets. Again, we have lots of nice, beautiful, incredible, interesting, exciting raffle tickets. We don't go through them one by one, but you can ask us anytime. $5 and probably $10,000 worth of prizes, so don't miss out. And you do not have to be present to win. So let's talk about what's coming up. As you might have heard, we had a few changes due to the weather, so we had to shuffle a few things. So tomorrow is Thursday, and at 1 o'clock you can join tonight's speaker. And after you hear him speak, I'm sure you're going to be intrigued to dive with him and also see how you can assist in recognizing if we have some of these problems. So be sure to register with us or see SEBA. Vicky's here as well. There's still plenty of space available. Don't miss out. 5:30. Our next presenter is famous Joe. Many people know Joe, and plenty of people looking forward also to this presentation. It's going to be at Liam's Cuisine out in the beautiful stars around the pool at 5:30. We will have the dinner specials soon, and you can let us know if you're also staying for dinner. We love when you support our sponsors. Look what we got this year. We've got cool hats. Bucket hats and baseball hats. You can't buy them, you have to earn them. Anybody who goes on any field project, you get a free hat. So another reason to go out and be a citizen scientist. Well, sorry. And of course, they are made with recycled plastic. So let's get started. Let's talk about tonight. Excuse me. I don't have this memorized, and Adam does not give me a teleprompter. Come on, Adam. Okay, so from amphibians to entire ecosystems, Dr. Pieter Johnson is revealing the hidden forces that shape our natural world. As a distinguished professor of ecology and evolutionary biology at the University of Colorado Boulder, Dr. Johnson studies how parasites and pathogens, often out of sight, have surprisingly big impacts on ecological communities and processes. His research focuses on the dual threats of disease emergence and species invasions using a powerful mix of experiments, large-scale field surveys, molecular tools, and ecological modeling. With over 200 peer-reviewed publications and recognition through top honors like the Packard Fellowship and NSF Career Award, that would be the National Science Foundation, that's big. Dr. Johnson has helped transform our understanding of how environmental change fuels disease. By shining a light on some of nature's smallest troublemakers, his work proves that parasites may be tiny, but they're infectiously interesting. Please join me in welcoming the other Pieter Johnson.
I'll take the little one.
Sorry. Yes,
Thanks,
you don't
Lynn.
need that.
All right. Well, thank you, Lynn. Can you guys all hear me okay? Yes. Excellent. Well, thanks for that introduction, and thanks to all of you for being here, and thanks to See and Learn for inviting me to be here. It really is an honor and a pleasure. This week, I'm supposed to be giving a midterm exam to my students in biological statistics, so they were really excited that I was coming down here instead, because now the exam is postponed till next week. And instead, I get to be here and talk with you guys about some of the work we've been doing. Fairly recent project, but focused on black spot syndrome, trying to understand the drivers as well as the implications for Caribbean fish communities. Before I jump into that, though, I just want to follow up on some things that Lynn's very nice introduction alluded to, and those are why we should care about parasites, right? Some of the interesting things about parasites that we're going to get into. And as a kid, I didn't grow up in the Caribbean. I grew up in Southern California. And so, like many kids in that area, right, I was captivated by the ocean. I was excited to hang out in the tide pools and go snorkeling and eventually diving and, get to hang out in scenes like this, to see things like sea bass, to see sharks, to see octopus, various invertebrates, echinoderms, crabs, all of these things just really captured my attention. And yet, really unbeknownst to me at the time, there's this whole other hidden ecosystem involving parasites, right? We can't often see them. They're beneath the surface, or in this case, beneath the skin or the tissue of our hosts. And yet there's all of these things in marine environments. Here, even in the rocky intertidal and the temperate reefs off California, you've got things like sea lice, we've got roundworms, we've got tapeworms, we've got microsporidians. Even this guy over here, this is a parasitic barnacle. Who even knew that barnacles could be parasites, right? But this one actually attacks a crab. It takes over the reproductive system, and it makes this crab take care of its own larvae instead of the crab's babies, right? And to me, as I learned more about this was just so fascinating. What are all these parasites? What are they doing, right? And why should we maybe care about them? One of the groups I want to introduce you to today are the trematodes. These are also known as flatworms or flukes. Probably most of you don't think about this group very often unless you're medical professionals. But I want to convince you as to why this group is interesting and some of the things that they do behind the scenes, the ways in which they can actually control what other species like their hosts are doing. And the key thing you need to know, if we just have sort of one takeaway from tonight, it's that these guys have these really complex life cycles. So they're not just a parasite of one species, but they actually require multiple species to complete their life cycle. So they're moving through all these different species in the food web, and that's the only way that they can survive and reproduce. So as a kid, one of the places that I would hang out in Southern California is this area over here. This is Carpinteria. It's near Santa Barbara, and it has these really nice salt marsh ecosystems, and I'd hang out there, splash around, try to find fish and other organisms. But what I didn't realize at the time is that this system is full of trematodes, right? Not ones necessarily that could infect me, but if we were to zoom in on this egret over here on the shore, what you would probably find is this adult trematode inside the gastrointestinal tract of this bird. This one is called Euhaplorchus. I won't test you on that, I promise. Just trying to give you kind of an example to introduce this group. But that adult worm is living inside the egret. And what it'll do is it'll release eggs when the egret defecates, and these eggs hatch and they'll infect a snail. In this system, those are typically horned snails, which are really, really common. And then kind of what happens in the snail is a little bit gruesome. The parasite will start from a very, very small organism. It gets into the snail, and then it starts replicating. And it keeps on replicating until it takes over all of the snail's reproductive tissue. Okay, so all of the gonads, all of the future offspring that snail might have produced have now become a parasite. And if you kind of want to get a sense for the scale of this, it's about 20% of that snail's tissue. So imagine if your leg had turned into thousands of these parasites and you just kind of had to lug this thing along with you, right? That's what the snail is up against. They no longer can produce their own offspring, and instead they just make parasites. So they turn into these parasite factories. This is the next stage that they make. It's called the cercaria. And those guys are actually free swimming. So they're looking for the next host, right? This is a multi-host life cycle. We've gone bird, we've gone snail. And now these cercaria are going to seek out and find a fish called a killifish. And they burrow inside the killifish and they form a metacercaria. And then they're just waiting for that fish to eventually get eaten by the bird, right? That's when the parasite gets back in the bird, it reproduces sexually, and it has offspring of its own that come out with the bird feces, right? So this is kind of a typical trematode life cycle. But here's the cool part, at least to me, right? I mean, the whole life cycle is cool, but these parasites aren't especially patient. They don't like to wait for these fish just to kind of randomly get eaten by the bird. So what they do is they infect a very particular part. No, it's not the eye. That's Diplostomum. That's a different trematode. Same type of thing. But these guys go for not the eye, but the brain. And that's the only organ they infect. And you can get tens, you can get hundreds, you can get thousands of these parasites all insisting right on the brain. Okay? And then what happens is that the fish that are heavily infected, they start acting weird, right? They have different behavior. They will do things like flash. They will do things like swim erratically or dart up to the surface. Or if the rest of the fish are all in a school, they sort of deviate from the rest of the individuals. And the net effect of that is to draw the attention of the egrets who come in and decide they're going to eat those fish, right? Those fish are drawing their attention. And some really cool experiments have shown that these heavily infected fish are 30 times more likely to get eaten by egrets, which is where that parasite gets to reproduce. It's where it gets to complete its life cycle, and it's how it spreads across the landscape. Yeah, question?
One egg produces dozens of larvae?
Yeah, so the question was, can one egg produce dozens of larvae? And yes is the answer. So one egg coming out of that bird, this is a parasite egg, not a bird egg, right? That will infect the snail, and then it'll just start cloning itself. So it'll go from a tiny, tiny amount of biomass to taking a fifth of that snail's tissue. Yeah, over here was first.
If we eat the fish, would we have the parasite?
Good question. So what happens if we eat the fish? Do we get infected? Not for this parasite. There are some trematodes that humans can get infected and get sick from. This is not one of them, and in fact, none of the ones I'll talk about today does that happen. Yeah?
How does the larvae find the snail?
Ooh, good question. How does the larvae find the snail? When it comes out of the bird, it gets into water, it hatches, and they actually follow chemical cues, signals, so that they can find the snail and then they penetrate it. Yeah. Yeah.
Do any other birds get involved besides egrets?
So it depends on the parasite. Some parasites are very specific about the bird. The question was, are there other birds besides egrets that get infected? And it depends on the parasite. For some of them, they're generalists. And for others, like the one we'll talk about later tonight, it's very specific. Yeah. Any other questions so far? Okay, excellent. So the kind of take-home that I want to push with this is that ecologists, scientists in general, are starting to appreciate that we can't afford to ignore parasites, right? That they may be small, they may sometimes be invisible to us, but they're doing big things. And this is just one example of starting to do things like, let's look at food webs with parasites and without parasites. And this is that same salt marsh system. Here, just showing you a food web of all the interactions involving free-living species. And now we can draw that same food web when scientists have gone in and actually added parasites. They've done all the work, they've figured out who's infecting whom. And what that does is it really changes the structure of the food web, not just because parasites are infecting free-living species, but even because they do things like free-living species will eat parasites, or parasites will break down and go into what's called the detrital food web and fuel primary production. So there's all kinds of interesting interactions that they're doing, and they change even things like our estimates of biodiversity, right? Parasites are a pretty substantial fraction of species richness in most ecosystems. And what's really wild is even that assumption that, well, parasites typically are small, they're only a very tiny fraction of the total biomass, the total abundance of organisms in an ecosystem. And now we're starting to figure out that that's actually not true, right? Because parasites are everywhere, they're infecting so many species, and they produce all of these infective stages every day. If we're to just look at the trematodes, our friends here, these flatworms or flukes, and you add them all up, you count all of the individuals, you weigh them, you will find that the trematodes, their total biomass, actually exceeds that of all the birds in this salt marsh ecosystem. So they weigh more than this sort of conspicuous group that we like to observe and go out and interact with. And what that means is that if as a kid, when I was hanging out here in this salt marsh in Carpinteria, if I could have seen those trematodes, they were hiding inside all of those hosts and in the bird and in the snails, it would have been the equivalent of a herd of 6 African elephants moving through this ecosystem, right? Something you would notice, you would pay attention to that, right? That would catch your eye for sure. So what does any of this have to do with coral reef ecosystems? What are parasites doing in coral reefs? The short answer is we don't know. There's so much about this ecosystem that is just poorly studied and poorly understood, especially when it comes to microorganisms and parasites. From some well-studied systems like the Great Barrier Reef, there's projections that if you're looking at all the reef fish species, and you're looking at all the parasites they have, and you kind of do some forecasting about how many parasite species there are, it's estimated that they would exceed the number of fish species by a factor of 10. So 10 times more parasite species than there are reef fish, of which there are a lot, right? And that's just for the fish, not even getting into invertebrates and so many of the other organisms in this system. What are they doing? How do their life cycles work? What do they even look like? All relatively unknown. And I would say this knowledge gap is really starting to hurt us. And that's particularly because of emerging disease issues in marine ecosystems and how challenging they are and how much they've increasingly threatened species that we care about or value. So today, finally after that long intro, right, I want to zoom in and talk about one example of an emerging infection for which the disease condition is called black spot syndrome. And what's interesting about it, or why I'm kind of pulling this one out, is because normally while we can't see a lot of the parasites or signs of infection inside a host, this one leaves sort of a conspicuous calling card. And this is an ocean surgeon fish, right? Normally these guys are just sort of a pale, light gray color. But here, when they're infected, they get all of these dermal lesions, or dermopathies, as they're called, that are associated with the infecting parasites. And that's allowed us to use some non-invasive approaches to also study this disease. And the history of this is fairly recent. Really about 2012 was the very first record I could find. That actually traces back to a blog post from the field station in Curacao. Karmabi, the research director there, posted that in 2012. Subsequently, some students were studying it in Bonaire. There was a report from Belize and a later one from Turks and Caicos. And then finally, the first published paper wasn't until 2016, so still fairly recently. And what these researchers are reporting is like, hey, we're seeing all of these fish with strange spots. Sometimes it's black. In some other species, those spots may show up as white. We don't really know what's causing it. We don't even know if it's all one thing or if it's multiple different things. Yeah, what is it? What's going on here? And really kind of serendipitously, in 2016, I was on a dive trip in Bonaire and happened to meet up with this guy, Zach Cole. He was working at the Bonaire field station, and he and his students had been studying this phenomenon. They've been seeing a lot of surgeonfish with these spots. They were trying to figure out what they were. And because I happened to sort of work on parasites, we got in the lab together, we started collaborating, and very quickly we were able to isolate this cyst structure and the melanin that's around it and open that up, and then found this trematode. It's called Scaphanocephalus. And it's super distinctive. It's really the only trematode that looks like this. And I'll show you what I mean in just a second. And it had never been recorded in the Caribbean before. Here's what it looks like when it's still inside the cyst. This one still alive, right? We've just removed it from a fish. And you can see the parasite, the worm, is moving inside. If you're to take this cyst and open it up, this is the guy that comes out of it. This is Scaphanocephalus, and it's the only trematode that has these wings, right? This sort of anterior widening of the forebody. No other trematode in the world looks like this. So as soon as we saw this, we knew exactly what we were dealing with, even before we sequenced it. And here's what he looks like when he's actually alive. So those wings move. Nobody really knows yet what the function of these wings are. Do they use it to swim through the water in an earlier stage? Do they use it to hug their mate inside the host and reproduce? We have no idea, right? But I think they're pretty cool. They're mesmerizing here to watch. The original name Scaphanocephalus translates to shovelhead. I see these more as the hammerhead sharks of the trematode world, right? They're really quite elegant. They're cool to watch. Really fairly beautiful. I could keep watching it, but I will move on so we don't stick on this slide. And it's a weird parasite. It's been described for 200 years, but nobody really knows much about it. The only thing we do know is that it uses these birds, right? The osprey. So this is an example where it is very specific. This is really the only bird that it will infect. It does infect a number of different reef species, reef fish species, like parrotfish and surgeonfish and others that we'll talk about in a minute. But we don't even know what the other host in the life cycle is, right? We were giving you that example with the salt marsh. It goes into the snails. Most trematodes use snails. A few use bivalves. But we still haven't even figured out what that host is. So a big part of this life cycle is still a mystery. So what I wanted to do tonight is tell you a little bit about what we have learned in the last few years that we've kind of been exploring these issues more directly. But this is very much a work in progress. So some of the things we're confident about, others we're still working on. And I welcome feedback, questions, your own observations, things that you care to share, and I'll try to give you a little bit of a perspective on where we're going. So we'll just briefly talk about which fish species are getting infected with Scaphanocephalus, which is one of the first things we wanted to figure out. Then we wanted to know, is this new? Is it increasing? Is it invasive? Or has it just always been around and somehow we didn't notice it? What's driving the infection? Why do we see more of it in some areas and less of it in others? And then most importantly, does it matter, right? What are the consequences, not just for fish that become infected, but really for the reef community or the ecosystem as a whole? So that's where we're headed. And I'll tell you that one thing I learned from teaching a lot of student classes is that I don't want to make it a mystery or detective novel, right? I'm going to give you the take-home messages right up front. Just make it easy. And the first take-home is that Scaphanocephalus infections are widespread and abundant in the southern Caribbean reef fish community, which is where we've been doing a lot of this work so far. And here I just want to give a shout out to some of the folks who've been really involved in this first part, Ramon Maluau and Lars de Horst, which is a former See and Learn contributor, alumnus. And these guys really good at catching fish and dissecting them. And what they found is that looking at a lot of fish in Curacao is that most of the fish species we examined, 29, I believe, out of 41, showed signs of infection by Scaphanocephalus, and that included the majority, or 63%, of all the examined individual fish. Now, what's pretty wild is just how high the infections could get. So up to about 1,000 metasarcaria per fish, and these are big parasites, right? So 1,000 of Scaphanocephalus is a lot of biomass, and nowhere else in the world has infections level this high been recorded, even in other places where Scaphanocephalus is known. Now this plot's a bit complicated. We're showing you the evolutionary phylogeny of all these fish, and then we're showing you the infection levels. But I'm just going to kind of boil it down and tell you that the fish that are getting most infected are these large-bodied herbivores like your parrotfish and your surgeonfish. They're the ones that tend to be most frequently infected, and they show the highest number of parasites per host. Not much effect of fish taxonomy or evolutionary history. Probably it's mostly about habitat, right? Are you in these shallow waters? Are you large-bodied? Are you feeding on algae? And for us, this was worrisome, right? Because these are the same fish that we are concerned about because of their ecological roles in reef ecosystems. We know they're critical grazers. They feed on algae that can sometimes threaten corals or reduce coral settlement by coral larvae. They play roles in calcium carbonate cycling and provisioning sand for beaches. So this gave us a bit of pause. This gave us a bit of interest to look deeper into what was happening here. And so part two, second take home, is that infections by Scaphanocephalus have really increased quite substantially over the last century or more. And I'm basing this off of work we've done going into museums, right? So we're going back to biological repositories, looking through fish that have been sitting in jars for 100+ years. In particular, I want to recognize Julia Piscawi and Ramon Malauau again, who spent a lot of time in multiple museums, but probably our biggest partner has been Naturalis Biodiversity Center in Leiden in the Netherlands. And the way we kind of tackled this is we were focused for this initial effort in Curacao and Bonaire. And so we looked through all of the old surgeonfish and parrotfish from 7 different species, roughly about 190 or so individual fish. And then we tried to go back to as many of those historical sites as we could, sample the same fish species from the same sites, and just compare, right? What are we seeing about patterns of infection? And Scaphanocephalus actually works very well for this because you can find the fish in the jars. The parasite likes to go for the fins, it likes to go for the skin, and then just shallow into the musculature. So we don't have to get deep into the organs. We don't have to destroy these valuable fish samples so that other researchers can use them down the road. We can isolate out the parasites. The challenge was you can't really get good DNA typically from century-old samples that have been sitting in formaldehyde. Some people can do it, takes a long time, it's really, really tricky. I'll spare you some of the details. But instead, we were using microscopy and histology to take these really thin slices of the parasites and then look for those unique traits associated with Scaphanocephalus, like those wings, but now they're all kind of folded up inside these cysts. And this actually worked better than I expected, which just about never happens in these kinds of projects. But here's a contemporary cyst of Scaphanocephalus. They have a lot of unique traits, not just the wings, but the nature of this cyst wall is actually pretty distinctive. And then this is a cyst from the 1940s that was really a dead ringer for Scaphanocephalus. So we were able to diagnose and identify parasites really well. And this showed us really 2 things. One, the infection's not new. It's been around at least since the 1940s. We didn't find any of it in our earliest samples, but that could also be because we had fewer fish from the period from 1905 to 1920. But the really big takeaway is just how much the abundance of infection has increased. So the percentage of infected fish has gone up by about 7-fold. And then the number of parasites per fish has gone up by about 50-fold, 49-fold in this case. Despite this being the same fish species, the same sampling locations, we tried to match things as well as we could. So evidence that the infection is changing through time, at least in the Southern Caribbean, right? We're working with other museums to sort of explore whether this is true elsewhere. Question? So the question is, has the parasite increased or do we have better technology? And I would say that this technology is good, it's okay, doing microscopy and histology, but we're using that same approach, the same sampling protocol, even in those samples from 120 years ago. Yeah. Question?
Does global warming have an impact on that?
Ooh, good question. Yeah, I think the question was about, what about global warming? Is that possibly contributing? And I think that's a good segue for this next question. We don't know for sure yet, but that's one of the things we're trying to get at. And I'll kind of get into that with this part. So the next part, which is still kind of in progress, some parts of it are known, some are still developing, but we're trying to figure out what's driving variation among reefs locally, and then more broadly trying to figure out what drives variation even among countries. And this is work for which Cheyenne DeWitt, a former master's student from University of Amsterdam, played a really big role. She's now at Reef Renewal in Bonaire. And what Cheyenne was working on was trying to develop these non-invasive methods, right? How do we go out and survey for infection without having to sacrifice so many fish, without having to dissect them? And so we're using GoPros that are on these kind of 1 meter long extension rods, and then doing transects at different locations, at different depths. And that allows us to sample a lot of fish and to get up surprisingly close to those fish because they seem to be less intimidated by the GoPro relative to us as divers. So here's what this looks like, right? This is one of our GoPros just on a 1 meter extension rod. Not terribly complex technology, but it does allow us to sort of get in there and, you know, get right in their face, and the fish are okay with it. Whereas if I was getting in there, maybe he would be a little less comfortable with that. So also kind of a plug, if any of you want to come out tomorrow, this is what we'll be doing. And I think, oh yeah, a little preview of kind of what this looks like. So this is just a video clip lifted from one of our transects. And again, what it allows us to do, especially in clear water, is get pretty close to these fish. You can see a lot of those dermopathies, those dermal lesions. We can observe a lot of fish. Some of those fish are lightly infected. Some of them are heavily infected. You can do these with different fish species as well. And some of them get pretty heavily infected, like that individual toward the top. And there have been some recent reports that in some parts of Saba, they're seeing more of this, according to Paul Sickle, who was here last year, the year before. So that's part of what we're trying to do tomorrow, is sort of follow up on some of those initial clues. Oh yeah, and good. I wanted to kind of show you what this would look like. So imagine you've collected that video, and now you're back on dry land. You're processing that video footage. Every time you see a fish, you pause the video, and then you're counting how many spots you can see that are consistent with Scaphanocephalus. So now I want you guys to count spots that you see on this fish. And then afterward, you're going to have to use your phone. Normally, I tell people not to use their phones during talks. Today, I'm actually going to have you pull it out so that you can enter in what your count is of the spots. So audience participation, take a second. See how many spots you see on this guy, and then we'll give you a chance with your phones to enter in those data in just a second. Okay, and as you're still counting, that's fine, but we're going to use this QR code, and probably everybody's used a QR code, but if you haven't, you're just opening your photo app, zooming in on that a little bit, should pull up a link from menti.com. You don't have to log in, you don't have to give them your email address, nothing like that. You're just going to try to put in your count for how many parasites or how many spots you see. And make sure this is working on my phone too here. Am I blocking you guys? Okay. Yep. Oh yeah, streaming audience. I apologize. So please feel free to also use this QR code. Hopefully it will work for you guys as well. Okay, have you guys got your counts mostly? Do you need Wi-Fi, or can you do it through data? Ah. Okay, we'll see what our people at home are doing. Okay, it looks like you can't see it on your screen, but on my screen, you guys are doing pretty well so far. One person put 4. That was way off. Sorry about that, whoever put that. But most of you, let's see what our average is. Yeah, you got it? Okay. Oh yeah, there's more coming in. We've got at least 18 of you so far, it looks like. Now I'll go back. Okay, good. You guys would be hired. All of you except whoever put that 4. I think that was you, Kevin. That was just wrong. But most of you got that the correct answer, if you will, was around 14. So pat on your back if you were pretty close to that. I sometimes did a circle with, multiple spots in it. Oh, you're talking about maybe an extra spot here. I would agree with you. That is almost certainly a parasite that's there. We sometimes try to be a little bit conservative. It's not a contest to find the most. It's more like if someone else is going to review after you, would they unambiguously be able to see a similar number of spots? And that's what this is all about. This is how our data works. Every video is processed by at least 2 people, and we compare their estimates to make sure the values are robust. And why this is so cool is because we know from fish we have dissected that the count of lesions, it's not a direct one-to-one, but it does give us a very reliable indicator of how many Scaphanocephalus are encysted in that fish. So again, just giving us a non-invasive way to go out, survey large numbers of fish, and figure out their disease levels. And that's what Cheyenne did. Question?
Yeah, once Scaphanocephalus persists.
So the question is about basically how long would it persist?
No, is there only a single Scaphanocephalus in one cyst?
Ah, good question. So if you're looking at one of those dermal lesions, is that one parasite? Sometimes is the short answer. I would say if you're seeing one of those fairly large, black, smoky abrasions, that's probably multiple parasites that are in there. So it's not a one-to-one, but you can still get pretty good indications of like, this is a really heavily infected fish, this is sort of moderately, and this is lightly or uninfected. There was another question too? Yeah. Yeah, so if the spots vary in size, is it a very good indicator? And I would say statistically, yes, it is. It's not going to tell you, there might be 100 parasites and maybe we only see 50 spots, but that 50 is sort of varying on a fairly consistent gradient to tell us about heavy infections versus light infections. Yeah. So the question's about, do we only count one side of the fish? Yes, but we have done other studies where we do count both sides with the video and then compare it to dissected animals, and it seems that there's not much difference between the left and the right side. Yeah. Any other questions? Okay. So these are some of Cheyenne's results from Curacao, right? And it shows a couple of interesting things. One is that a lot of the fish are infected. About 70% of ocean surgeonfish in Curacao along the leeward coast are positive. And that it is increasing as you go from the east side of the island toward the west side. If any of you have dove East Point, Curacao, there is almost no infection at all here. It's very hard to find an infected fish. Why is that? What are some of the drivers of this? Well, it seems that the hot spots are the areas that are fairly calm with low wave intensity, and areas that are shallow and have high nutrient runoff. So we're seeing a higher signal for nitrogen isotopes that are associated with anthropogenic activity in the algae. So that could influence the snails that are still unknown. It could be influencing the fish, but that's how we can sort of predict where some of these hotspots are. And then what we're working on now that we've been really trying to dig into more is, so how do we kind of expand this approach? It's one thing to do this on reefs in Curacao. It's really valuable, but it's local, right? It's one island. So how do we up in scale geographically? And here we've been partnering up and using iNaturalist, which, as probably all of you know, has now become one of the world's leading community science, citizen science repositories. You can upload your observations, you can geotag things, right? And we're just zeroing in and focusing on the reef fish, like this ocean surgeonfish. And I kind of took pity on you guys here. I didn't make you count this one. He's got 98 spots, so that would have taken us a little bit more time to work through. But this has been really interesting. It's taking some time because we're looking through a lot of different fish species. We have this kind of student army back in Colorado that's being led by Riley Klein. And for these 20 species, now we've almost finished going through 42,000 images. And this has been, to me, really interesting because, yes, you're seeing infection in the surgeonfish, in the parrotfish, but also in things like filefish, right, that show a lot of infection in the fins especially, in sand tilefish and in some species of razorfish where the skin is pale enough that you can really see those dermopathies quite clearly in contrast. So this is still in progress, but here's kind of some results just for ocean surgeonfish, so just one of those 20 species. And why I like this is it starts showing us some of the hotspots at the scale of the Caribbean. So yes, the southern Caribbean seems to be where we find the most black spot syndrome, Curacao, Bonaire, a little bit lesser so in Aruba, but that general vicinity. But, oh yeah, sorry, question.
Does this reduce the lifespan?
Yeah, so the question's...
Also like the quality of life.
Yeah, the quality of life. So the question is, does this reduce the lifespan of the fish? And what about the quality of life for the fish? And lifespan, we don't know, but we do know that it damages the fish, and I'll show a little bit on that. Quality of life, if I had to guess, I would say yes, because the parasite is damaging the fins. It is causing hemorrhaging under the tissue, and it's a significant amount of biomass. They're showing more energy expenditure, as I'll show you, so that would be my guess. Yeah?
Are you doing anything with the fish moving around, migrating?
Yeah, so the question's about what are we doing with respect to the migration of fish. It's a nice little gecko there grabbing that bug. I like that. We are looking at fish movement, especially with respect to schooling. And we're finding that infected fish are way less likely to school than the uninfected fish. And we're trying to figure out why that is. And certainly the schools of surgeonfish move around way more than the solitary individuals. Yeah. So what I was going to say, oh yeah, sorry.
If you look at this graph, you see the spot counts per fish.
Yeah.
So it's not the observation, or how many observations have been?
Yeah, each fish is a dot in this case. So there's 4,671 total surgeonfish that we could use the images for. And so all of the individual dots are individual fish, and then the size and color of the circle tells you how many spots per fish. Yeah.
Does the water temperature have anything to do with it? How high up does this actually being found?
Yeah.
Is it found off New York?
So the question is, what's the role of water temperature? And I would say very important. So in our statistical models, average water temperature is really one of the strongest predictors of where we see infection. And it definitely does not extend farther north than around parts of southern Florida. We don't see it up in Bermuda, for instance. We don't see it anywhere along the US coast. It's definitely a tropical parasite. Yeah.
But with water, with the warming of the water,
So into the future,
it's going to migrate and probably into other fish.
yeah, so the question is about what's going to happen as the water continues to warm. Could that change the parasite's movement? Could it move farther north? It's certainly possible. And the osprey do move the parasite pretty far north. So I imagine it is water temperature that's limiting how far the parasite can establish. Yeah. So Southern Caribbean, we have a big hot spot, but I'm also intrigued by the barrier islands of Belize, the Yucatan around Cozumel, and then these sort of lesser hotspots near Turks and Caicos and Bahamas. So far, the biggest predictors have been water temperature, and especially like stable water temperature, right? Where it stays pretty warm all year round rather than seasonally getting cooler. And then, in addition, osprey abundance is a very important predictor, as you would expect. And osprey, as I'm sure most of you know, are highly migratory, at least the North American osprey. These guys are tending to breed up in the United States and Canada right? So they're during the spring and summer, they're up north. And then as things start to cool down and we get toward the winter months, that's when they're all funneling down through the Caribbean, and especially making this movement down to the northern coast of South America. And that's when they're pulling through Bonaire, they're pulling through Curacao, probably explaining at least some of that hotspot along with the temperature. Now, some of you guys are birders, so I'm sure you can tell me, what about Belize and the Yucatan, right? I showed you hotspots there, but I don't see many of these migratory osprey hanging out in that area. So what's going on over there? Why might we see so much infection in that area? Resident osprey, right. And which ones are those? The subspecies that is the Caribbean osprey. Exactly right. They're residents, and they spend all year round in this area, especially on those small islands around Belize or in the Yucatan, and they don't migrate, right? They're just breeding in those areas, and they're there in wintertime, and that's exactly where we're also seeing pretty high infection. Yeah, Lynn.
Are your statistics skewed at all? Are you relying on citizen science for the photographs? So are they higher, are there more photos in more developed touristy areas? Or have you sampled fish in more remote areas? And is that a difference? Or are they impacted by development?
Yeah, so the question is, well, using citizen science, what are sort of the limitations about how representative this might be of what's kind of actually happening out there, or how much representation there is for some geographic areas? And I would say, yes, we have investigated that. So we've looked at areas where we've done transects by researchers, compared that to iNaturalist. And in the areas with good coverage, I would say the estimates are very solid. Where we suffer is that there are some locations, say Cuba, for which we have way fewer observations on iNaturalist than we do in Bonaire, right, or Cozumel. So there are gaps on the map, but I don't think the data points that we have on the map suffer from that kind of bias. Yeah, great question. And I won't talk about it today, but one of the researchers in our lab, Dana Calhoun, her main work is on looking at the osprey themselves. So she's working with bird rehabbers, veterinary scientists, zoos, and when osprey do unfortunately die, right, in various locations in the US and throughout the Caribbean, she's getting those carcasses, she's finding their parasites, and using population genetics to try to figure out where are these parasites going, how far north do they go in the US, could there even be multiple species of Scaphanocephalus in the Caribbean, which is still something that we're trying to figure out. So that work is ongoing, and I won't dig too much into it today, because the last thing that I just want to talk about for a few minutes is exactly what you guys asked about, right? How does this affect the individual fish or the fish community? What we do know is that the fish that are heavily infected have lower body condition, right? They have lower mass relative to their size compared to uninfected or less infected fish. We know they suffer from damage to the fins. They exhibit hemorrhaging based on histopathology. They show oxidative stress damage and altered immune reactions based on various assays that scientists in the Mediterranean have been using. And what we're trying to do is really extend those individual fish level effects, those sort of physiological effects, to get at shifts in behavior that can influence things like grazing capacity of either the individual fish or the fish population. And this is some work that's really being spearheaded by Anna Poślednik, a PhD student. And some things we have already been able to document, like heavily infected parrotfish and surgeonfish spend less time grazing, which is worrisome. They spend more time fighting or engaged in antagonistic interactions with other fish. They're way less likely to school for the surgeonfish, right? We're still trying to figure out how that works or what's driving that. And they do show evidence of higher energy expenditure relative to uninfected fish. So they're working harder to swim and to graze. And what what Anna's doing and why she has this sort of bizarre chessboard and stuff is that she's using this really cool stereo camera system that's calibrated between the two cameras so you know the distance. And then she can reconstruct all the movement of the fish in 3 dimensions, which is why we have to calibrate it. But it's super time consuming. It requires a supercomputer to process all of the data. So she's back in Colorado, very busy working through all this. So stay tuned for more on that. I did think I would show you one of the experiments she just did in Curacao about a month ago. Again, data are still being processed, but I thought it was such a clever experiment that she did. She was trying to look at how infected and uninfected fish forage in areas that are risky, like this sand flat. There's very little refuge here, versus areas that are close to the reef, right, where fish can hide, and it's known that they're a little bit more comfortable foraging in those areas. They're less at risk from a potential predator. And then she's got this seaweed that is deployed at different vertical depths through the water column, again, sort of replicated in the sand and by the reef. And then she's looking at how fish come in and feed on these different algal pieces. These are all doctor fish, all of which are infected to some degree or another. And again, trying to get at what is the grazing capacity of these infected versus uninfected fish across different reef sites. So again, stay tuned. We'll have more results on that, I hope, soon as Anna gets through it. But what I kind of hope that I've showed you in terms of where we are right now, we know now that black spot syndrome is caused by this trematode, Scaphanocephalus. It's a fairly obscure parasite, never previously recorded in the Caribbean. And at least in the southern Caribbean, it's very widespread throughout the fish community, and especially in our large-bodied grazers, the surgeonfish and the parrotfish. Based on these historical reconstructions of what has happened through time using museum samples, we know that infection has gone up in places like Curacao and Bonaire, and quite substantially so. And we know that among reefs, we tend to see the most black spot syndrome in these calm, shallow areas with high nutrient runoff from human sources. And across the Caribbean, still very much in progress, but we're seeing more of the disease in these stably warm latitudes, right, where water temperature stays high, and in areas that have large numbers of osprey, either migratory or residential. This is the area where we're still most busy. What is the infection doing to the fish? We know it damages individual fish, but does that matter to the population or to the ecosystem in terms of the grazing potential that that fish community has? And we're getting there. We're starting to see links to changes in behavior, but I'd say that part is still in progress. Now, just going full circle, if any of you are paying attention closely, I still haven't addressed one of the biggest mysteries for this parasite, which is, what the heck is this missing host, right? This is a big piece of the life cycle that we still don't understand. Most likely it's a snail, about 99% probability. There are some trematodes that use clams and other bivalves, but we don't know which one. And we've spent a lot of time trying to figure this out over the last 3 years. We've gone through nearly 50,000 snails and other mollusks trying to find this parasite. And what's cool is we've found an enormous number of trematodes. All of these are those Cercaria stages. That's cute, right? That guy? Yeah. Smiling at you. These are all Cercaria stages of different trematode species. Many of these are undescribed species. Some of these are undescribed genera, for which scientists really don't know much about, right? There's been very little work on these parasites in regions like the Caribbean. Tons of cool science to be done here, but none of these are Scaphanocephalus. We've sequenced them all, done 28S, done COX-1, we've done morphology, and none of these are the parasite yet that we're looking for. So the treasure hunt continues. And we're trying to work now more with scientists from other parts of the world where Scaphanocephalus has been observed. And puzzling things are, there's been a lot of research coming out of the Mediterranean since they estimate in 2016 the parasite first became observed and has been emerging in their commercial razorfish industry. They harvest pearly razorfish, and they hadn't seen it before 2016, and now they're seeing more and more of it. So hopefully through teamwork, we'll kind of solve this part of the mystery. And I wanted to end just by, again, emphasizing very much work in progress. There's still things we're trying to figure out, and we benefit from any data or observations that you care to share, whether they're here from Saba or whether they're from elsewhere in the Caribbean or the world. So feel free to email me. Feel free to chat with me later. Join us tomorrow if you want to take a look for any infection in Saba and kind of see what we can find. But otherwise, yeah, I just want to thank you guys for hanging out. Thanks, See and Learn, for inviting me, and happy to answer questions. Yeah.
It sounds like there are several Schiphanocephalus species. Do they all use the osprey as a stage?
Yeah, so the question is, okay, there are multiple Schiphanocephalus species throughout the world. Do they all use osprey? Mostly yes. There is one record from 1916 in Australia where they reported it from an Australian sea eagle. There's some debate as to how good their identification was. Sea eagles can look a bit like osprey, so it might have been an osprey. The other records from Australia are all from osprey. So it does seem to be this remarkable specialist of osprey. Yeah?
And does it have any adverse effects that you know of on osprey?
Good question. Are there any potential adverse effects on the osprey? I would say we just don't know yet. There can be some evolutionary arguments that it shouldn't hurt the osprey because the parasites depend on it for dispersal, but I would say that's sketchy, right? We don't have direct evidence to support that. Yeah. Yeah.
Is it possible there's nothing else in the life cycle besides the fish and the osprey?
So is it possible that there is no other host in the life cycle, and it goes directly from the osprey and then infects the fish? I would say, working in science, anything is possible. I would say it's highly, highly improbable because the size of the egg that comes out of the osprey is about 20 to 30 microns. And then the size of the metasacharia, even the smallest ones we can find in the fish, is already about 600 microns. So they would have to do some massive amplification, and those stages are all non-feeding. So we'd be, yeah. So I would say probably not. Yeah.
What is the black spot doing, basically? Is it taking over the reproductive organs, or what's it doing in the fish?
So what are these black spots doing in the fish? And I didn't talk about that much. This is a melanization reaction that involves the immune system. But I would say the coolest idea, or the one we're most interested in testing, is that this makes the fish more conspicuous to an osprey. Right? Osprey are remarkable visual hunters. They're flying over that clear water. They're looking down. They're trying to figure out a fish to attack. And if these guys are swimming shallower, or if they're flashing, and if they have these spots, does that make it that much easier for the osprey to dive down and grab that fish? So that's one of the things we want to try to figure out. Yeah. Any other questions? Okay. Thanks, guys.
Adam? Hello? Thank you, Peter, for putting this obviously very complicated work of yours at a level that we feel like we can all understand. Doesn't mean we could repeat it tomorrow. So everybody, thank you for coming. If you haven't tried the chili, you absolutely should. It's fantastic. And the bread. If it's your first See and Learn experience, hopefully you saw how awesome it can be with a dynamic speaker. Please, by all means, come to Emily or me. Tell us that you want to dive with Peter tomorrow, with Bob on Friday. And by all means, let's see you at our next talk on Friday at Liam's Cuisine. And check the posters, our website, social media, and we look forward to seeing you more. Thank you.